Hello everybody, welcome in today's video. Last time in the first video we have stopped in this slide, so we are going in today's video to continue in the introduction to the neurodegenerative disease. So in this slide we will talk about how the healthy neurons are transformed to the disease and neuron in the case of Alzheimer disease. So as it is shown here in the title of the slide, so from healthy to disease neuron, the role of microtubule. Those microtubules play an important role in the maintaining of the structure of the neuron. So here I will present you the top form of the healthy neuron and the neuron that are attained by the Alzheimer disease. So here in this image here, it is represented the neuron with microtubule, with tubulin dimer, and here with macrotubule and the cross section here. Here is an overview of the same one, here, but here is another view. Let's say here as it is present here, the here in the yellow color represent the beta tubulin, and in the red color represent the alpha tubulin, and here this black one represent the tau protein, and this one represent the kinesin. So the kinesin, what is the kinesin? The kinesin can be considered a molecular motor that transport vehicle along the microtubule like a railroad. So for example, you can consider this one as in the management when they want to transport something inside to the outside. They use some let's say the how to say it, as it is like a railroad, and they use it as a tool to transport a thing. And in this case, in the biological system, the kinesin play an important role as a transporter vehicle along the microtubule, and this microtubule can be considered as a railroad. Now coming to the formation of neurofibrillary tangent. So here is a healthy neuron, in which healthy neuron we will talk in the normal case, and in the healthy microtubule, and in the Alzheimer microtubule, as it is shown in the image here. The presence of the microtubule has a result to the stabilization of tau protein here, as it is shown by stabilization of alpha tubulin and beta tubulin, which form a dimer, and those dimer form what we call a macrotubule, and those macrotubule are stabilized by the tau protein. As I already said, that those tau protein play an important role in the maintenance of the structure of the microtubule. Now, what happened in the Alzheimer disease? In the Alzheimer disease, those tau protein are not stabilized. We will see all those in detail, how the mechanism by which those tau protein are destabilized. So we will see this in the chapter of Alzheimer disease. Here it's a general introduction. So what happens? Here in the Alzheimer disease, the tau protein are not stabilized, and as a result, the macrotubule are not stabilized, and the generated tau protein are accumulated, which led to the formation of the tangent tau protein. So the formation of neurofibrillary tangent acts as roadblocks, and as consequence it impairs axonal transfer by the kinesin. This one here, we know that this kinesin to transporting it need like a road, but in this case when there is a destabilization of tau protein, there will not be a road by which kinesin can transport. So with that will lead to the loss of synapse and memories dementia in patient, and we can talk about the forgetfulness. We spent a lot of time in this slide, so let's move to the next slide. So here the age-related evaluation of tauopathy in Alzheimer's disease. Here is this image, or this schema here represents the early Alzheimer's disease with a neuron that is normal, and this red color represents the high tau level, and here in this part tau level. Those in green represent the A beta protein, and this in yellow represent the A beta induced toxicity. So the disease progression, what happened? The phosphorylated tau and the endogenous tau, the dendritic tau level. Here is the early disease. Here is the later late in Alzheimer's disease. What happened is in the late of Alzheimer's disease, in the late of Alzheimer's there will be a high tau level as it is shown here, and less, and here in this part there will be a low tau level, which will have as consequence the destabilization of the road in which the kinesin transport protein, so there will not be this part of the axon will lead, because there will not be generation of protein, and the vesicle here, we know that the vesicle for transmission are located here, and those vesicle need to have a metabolite which come from the neuron center, and as consequence there will not be the synthesis of neurotransmitter, and the non-synthesis of neurotransmitter will lead to the non-communication with other neuron, and as a result this neuron will not have either connection with other neuron, as a consequence there will be the death of the neuron by lack of connection with other neuron. So for the possible pathway, let's talk about the possible pathway from the extracellular beta amyloid protein to neurofibrillary change. So here, as it is represented here, we have the protein, prion protein in the yellow here. We have the Alzheimer A beta oligomer here. We have the physiological glutamate in blue 5. We have the post synapse. We have the fin, the loss of synapse, the pito, and this is a mechanism. So what happens in the normal case, this pathway are okay. But what happened in the Alzheimer's disease? In the Alzheimer's disease, it is characterized by the accumulation of A beta oligomer. Those A beta oligomer will fixate in the prion protein. Fixation in the prion protein will inhibit the physiological glutamate or the melat, or let's say the mutation in the prion protein can result in the neuropathological disorders such as mad cow disease. But this protein also serves as a docking site for the amyloid beta protein in the case of Alzheimer's. More, A beta here binds to a protein and the prion protein, and as a consequence stimulation of mGlu5 glutamate receptor, and as a consequence of the stimulation there will be morphine kinase activation. The morphine kinase activation led to the loss of synapse, and at the same time the degeneration of the p-tau protein, and as a consequence the neurofibrillary change of Alzheimer's disease. So the immunotherapy technique that would utilize antibody to target the A beta binding site on the prion protein are currently being researched. So let's move to the next slide. Here we'll start a general introduction to the Parkinson disease, but I don't know if we are going to continue in this video or to start in another video, but let me just give a little word about the Parkinson disease. So Parkinson disease, it is a progressive neurodegenerative disease that is discovered for the first time by the Jim Parkinson. It is an American doctor. So this disease is characterized by the degeneration of dopaminergic neuron in the substantia nigra pars compacta. Here is in the normal case, and here is in the Parkinson disease case. And this pathway here represents the negrostriatal pathway, and you see here is the frontal cap of the brain, and here you see the caudate nucleus and the putamen, the internal globus pallidus, external globus pallidus, and here is the thalamus and the hypothalamus, and in the same part here you will find the amygdala and the hippocampus and the mammillary battery in this patch, but those are not important for us in today's course. We will focus on the putamen in the caudate nucleus and in the substantia nigra pars compacta. So here, this image here represents the Lewy body like synuclein and the ubiquitin, which are characterized. We can find this protein in the brain of the Parkinson patient. So Lewy bodies located into the dopaminergic neuron in patient substantia nigra. Or they say this Lewy body, we will find it in the cap of the substantia nigra pars compacta of patients that suffer from Alzheimer's disease, and we will see all this information in the child video when we are going to talk about the different characteristics of Parkinson disease. Now coming to the Parkinson disease as a neurological pathway drug target. So here is the schema of a brain in which here the prefrontal cortex, the occipital cortex, the parietal cortex, and the temporal cortex here, and here is the cerebellum. And here it is located, here is the striatum, in this part, with the caudate nucleus in the same part here, and also the globus pallidus internus and the externus and the internal nucleus also, and here is the fourth ventricle here, and the lateral ventricle in this part here, and here is the white matter here, and here is the gray matter. But as you already seen in the slide before, we will focus on the striatum because Parkinson disease affects the dopaminergic neuron in the substantia nigra. So here is the pathway between the striatum and substantia nigra. So here the pathway from the normal striatum. So here is the substantia nigra, which project negrostriatal pathway to the striatum by dopaminergic neuron. Those dopaminergic neuron can either stimulate or inhibit by dopamine D1 receptor or inhibit by dopamine D2 receptors. So this dopamine can stimulate the GABA. Even if they stimulate GABA is an inhibitory one, acetylcholine is an excitatory one, glutamine is an excitatory neuron. So here in the case of the Parkinson disease, here we lost the dopamine neuron which came from the substantia nigra, and as a therapy we use the L-DOPA or dopamine agonist in order to stimulate this neuron here. And we saw that we can also use the MAO-A beta or the COUNT. The MAO-A beta and MAO COUNT L-DOPA dopamine agonists are medicine or that are used in Parkinson disease in order to limit let's say the manifestation like tremor, akinesia, bradykinesia in the Parkinson disease. I will stop the recorder in this slide and let's see you in the next video, in which we are going to talk to finish this part, and then we will start with the Alzheimer's disease in detail with mechanism, and it's different. In other video we'll talk about Parkinson disease. So thank you for your attention, and don't forget to subscribe, make a like, and share, and also